All right, for our next part of our unit on cells, we're going to talk about mitosis and the cell cycle. Okay, so why do we need cells to divide? There's usually 3 reasons for a cell to divide, which means we need more of them. So for growth, so obviously as a baby, you needed to get more cells than you'd had then for you to get the size you are now. Development, so when you started out as a single egg and sperm cell joined, you had to develop from there and keep dividing those cells for that to get bigger. And then also repair. So when you cut your skin, you need to grow skin cells back in order for that to repair itself. So we need cell division in order for that to happen. Okay, so we have something called the cell cycle. All right, cells sit through a certain cycle throughout their life of their cell. Most of it is in our growth phases, which if you see over here, we have a G1 and a G2 phase. These are our growth phases. So sometimes cells need to keep growing after they've divided in order to get bigger. We're going to talk about this a little bit later. We have a phase called S phase. That's DNA replication. So we're going to replicate our DNA. And both of these types of phases are in what we call interphase. Okay, so this is the phase in between cell division that this cell is just growing or it's replicating its DNA. All right, the other phase we are really concerned about is the M phase or the mitosis phase. Okay, this deals with mitosis, and what we're going to talk about is cytokinesis. Okay, so this is how cells divide in this M phase of the cell cycle. Okay, so what happens during cell division? So we need to, when a cell divides, we need to take the information that cell has and pass it on to the new ones. So we have to have a complete copy of the genetic information. So we need to make a copy of our DNA and make sure it gets to both of those new cells perfectly. All right, we also need to, as we talked about, we have all these organelles in cells. They need to also be passed on so that the cell can divide. Right, so we need to have not only DNA passed on to both of our new cells, but we also need all of those cell organelles passed on to both of the new cells that we get when a cell divides. Okay, so DNA, what happens to it? So during that interphase part, which I mentioned, in between those 2 growth phases, that S phase, that's when we are going to double our DNA and the organelles. Okay, so we have to make double if we're going to split the cell in half and for both cells to have all of it. All right, the DNA, while it's in interphase, is kind of just sitting there nice and uncoiled in a form we call chromatin. Okay, this is an uncondensed, not coiled up form of DNA. Okay, so that nice double helix you've seen, it kind of just is sitting out there nice and loose. Okay, so when we get to start dividing, our DNA, if you think about it, if you had a ball of string that was all unwound and you try to pull it apart into 2 pieces, it's going to get tangled, messed up, something's going to break off. So DNA actually protects itself and it condenses into what we call chromosomes. Okay, so these are tightly wound DNA around a center part that's called a centromere. Okay, and it's very important for these DNA molecules to get up nice and wound tight so that nothing breaks off or we don't lose any information. Okay, so this is kind of what it looks like. This is DNA in its helix form in number 1. It'll start wrapping around proteins, which we call histones, and that kind of helps organize the DNA, and then it starts condensing. This is that little centromere center part right here. So these are long tails, and then they start folding up like this, and then we get our nice chromosome right here. Okay, so this is just another picture of it. So if you want to see it in full form, our DNA double helix, which you've seen a picture of already, around these histone proteins, wrapped up in bundles, wrapped up in more bundles, wrapped up in more coils to make a chromosome. Okay, all to protect our DNA. Okay, so what is mitotic cell division? So we had that M phase I talked about. Now, what is it? So this is what normal, regular body cells called somatic cells undergo in order to divide. Okay, so in our body we have somatic cells, and then we have our sex cells, our sperm and egg cells. They go under a different division, but every other cell in your body is going to go under mitosis. Okay, so where this single cell is going to divide into two identical, perfectly identical daughter cells. Okay, so this is just a cool little picture. This is a actual a cell dividing, and what they do is scientists can use fluorescent proteins, which is why you have that bright green, bright blue, and bright pink, and take pictures of it under a microscope of the different parts of the cell. So this is the nucleus with chromosomes in it, and we have other stuff out in the cytoplasm, and then this is our cell membrane. Okay, so the three major parts of mitotic cell division that you need to care about. Interphase, which is where we have the cell preparing to divide, and we duplicate our DNA and organelles. Mitosis, which is just worried about dividing the nucleus and the chromosomes. Okay, so we need to get all that DNA out. And then cytokinesis is the cytoplasm division, or dealing with making sure our mitochondria and everybody gets into both cells. Okay, so mitosis itself. There are four parts of mitosis: prophase, metaphase, anaphase, and telophase, with cytokinesis at the end. The way to remember this is PMAT in order. So prophase is this number 1 down here. Metaphase is number 3. Anaphase, telophase, and this will be cytokinesis on this last one. Okay, so our first part of our mitosis phase is called prophase. All right, what happens in this phase is chromatin, which is our uncoiled DNA, is going to condense into chromosomes. So if you see these little X's over here, these are our condensed chromosomes, right? So they're nice and organized. We are going to have, you heard about the organelle, the centriole? Those are these little orange guys over here. Okay, what they do is they send out these spindle fibers, which are going to come out, these little blue pieces over here, and they're going to come out and start growing to try and attach to these chromosomes. All right, the other thing is all of our DNA is inside the nucleus of the cell. Okay, and the nucleus has a nuclear envelope. In order to get this DNA out so we can split it, we have to break this envelope down. Okay, so that's going to break down in prophase. Okay, now that we have everything out of our nucleus and we have our chromosomes attached to our spindle fibers right here, then we need to make everything meet up in the middle so that we can divide this equally. All right, so it's going to meet up at the equator or the middle of the cell, nice and lined up like that. So actually this is another one of those dyed fluorescent cells, and you can actually see all the little chromosomes right in the middle here. And then these bright green things are the spindle fibers attached to the centrioles. So our third step is anaphase. So all those chromosomes were right in the middle, and remember they were attached at a centromere. So both spindle fibers on each side are actually going to grab one half of the chromosome and pull it to either side of the cell. Okay, these halves are called sister chromatids. Okay, they are exact copies of each other. Okay, and how these spindle fibers pull these apart is that they actually shorten. So instead of growing to go connect them, they're going to shorten now to pull them apart. All right, so this is actually a dyed cell version. So this is still the DNA up in the sister chromatids now, and then our spindle fibers pulling it to either side of these cells. All right, the last phase of mitosis is telophase. Okay, so we already had our spindle fibers bring everything over to each half of the cell, and these are going to start to disintegrate, and they're going to unattach from the sister chromatids. All right, now we remember the DNA was originally in the nucleus, So we need to remake those nuclear envelopes in order to keep our DNA where it's supposed to be. And then those chromosomes or chromatids that separated, all of those are going to go back to their unraveled state and start there. If you want to see, this is another one of that same picture from earlier, died, and you can see that the cell is almost separated but not quite. Okay, so this very last part of cell division is called cytokinesis, right? So we have separated the nucleus and DNA and divided them, and that was the whole point of mitosis. So in order to finish off the cell division, you need cytokinesis, which means we're going to separate the cytoplasm and the organelles equally between these new cells, right? So if you look over here, there's a mitochondria on both sides, there's some endoplasmic reticulum, and there are centrioles on either side. Okay, so this is that very last step when cells totally divide off and split up their cytoplasm. So if you can see, these are another set of dyed cells, and that very last part is being split off, and these are going to be two exact new daughter cells. All right, so that's cell division, and that will be the last thing that we cover for our unit on cells and cell membranes.